this video, Dr. David Francis from The Ohio State University talks about prediction using genome-wide selection models. He begins by talking about the population being used in the analysis. This topic was discussed in details in the previous lecture, which you can click the link at the bottom of the screen. Then, he talks about the importance of including check plans to genotype. And finally, how to predict individual future performance based on only the genotypic data and obtain selections using our software.
Still going to focus on the bacterial spot data. And what we've done up until now is that we've talked about selections from our initial population that were used as a training population. So we made some selections. We had that population distribution. We decided that at k is equal to 1, there were individuals that we were going to keep. So we take those selections, we intercross them, and then we self-pollinate them. So now we have a new population. It's related to the old population. Essentially, the best parents were chosen, intermated, and then selfed. So what we're going to do with those is we're going to grow them out in seedling trays. We're going to collect tissue and extract DNA. And then we're going to send that plate of DNA to a service lab and ask them to genotype this collection with the same markers, exactly the same markers that were used to develop our model. So now what we're going to get back is a matrix of genetic information. And we want to predict the performance of our individuals based on only genetic information. And this, by the way, is the thing that's really exciting about genomic selection. It's the potential that we could predict an individual's future performance based on only genetic data. That's what we're after here. Okay, so we'll have only genotypes for this new population. So again, I'm walking you through that. We've made our selections. We've sent a plate, and I'm only giving you one plate versus marker data. We're using a competitive allele-specific primer assay, and the genotypes are getting sent back to us essentially in a spreadsheet. We do include some checks in this plate, and we evaluate those first to make sure the phase of the marker data is the same as the phase in the training population. And this is important. And the reason that this is important is that if I develop an assay, say on the Illumina platform, and then I want to convert that assay to another platform, they may decide to reverse the primers because they're going to get a better assay. But if they reverse the primers, they're going to then call that SNP in the opposite orientation. I'm actually not very good at like flipping my thinking over and getting this stuff right all the time. It's easier for me to give up one well on a plate to a check so that I can verify that the data that I'm getting back is actually in the same frame as my historical data set in my program. If you're getting SNP data from an Illumina array, the person using Genome Studio gets to select how they call that SNP. Am I calling the SNP based on the design strand of the primer, or am I calling it versus a reference genome? And you can have a project where everybody agrees we're calling it based on the reference genome. And then the reference genome changes. And you're doing what you agreed to, but the data set from 2012 to 2016 is not the same as the data set after 2016 because the reference changed. Right? All that has to happen is somebody has to decide we had the whole telomere of chromosome 11 reversed and we need to flip it over. And now you have 1,000 markers that are being scored differently. And if you don't realize that, if you don't figure that out, all of the population genetics that you do is going to have this division between, the first couple of years of the project and the second couple of years of the project that aren't real. The other thing about including a check, if you put that check asymmetrically in your plate, it's a way of guarding against plate flipping errors. I'm putting my DNA in this plate, I'm sealing it up, and I'm sending it to a service lab. If that service lab isn't careful with the orientation of my plate, they may randomize my data. How am I going to figure that out? Right? They're going to insist that they do everything the same all the time. But you and I know that sometimes you walk in and you're tired that day, and it's really easy just to invert a plate. Now, so how are you going to know that? Every single plate should have a check in it. Your job is to realize where the common mistakes are and to account for it. All right, so having a check in your plates, an asymmetrical check in your plates, accounts for scoring differences and mechanical errors in how that plate is oriented. It's very important, right? It may seem trivial, but it's very important, right? So we've sent off our plate, the data have come back. The first thing we do is check to make sure that the checks are evaluated, to make sure the phase of the marker data is the same as in the training population. This is where we are in the process. We have genotype data and we need to make some selections. This is what it looks like. So we have this marker estimated breeding value that we're going to read in. We're going to call it E. 397 markers by one vector of values. Right? This is our model. The next thing we're going to do is we're going to read in our plate data. We're going to call that selection, and so the dimensions of that are 96 by 397. Okay, so 96 individuals, 397 markers. Okay, so what we want to do then is multiply these so that we end up with a 96 by 1 vector that is going to be the value of each individual from our plate. All right? So we have to have, these have to be the same number for us to be able to do this operation. And we're going to end up with a 96 by 1 vector. And that is now the prediction for our new genotypes. So this is coming from the output of either RR-BLUP or DLR. Okay, now here's the thing. If you don't have exactly the same markers, you can't do this. And so this is where genotyping by sequencing becomes somewhat problematic. If you have a set of markers that you've used for your training population, and then you send some new material away to be genotyped by sequence, it's going to come back with an overlapping but different set. So if my training population has this set of markers, and my selection population has this set of markers, I can only use this set of markers, which means I'm going to have to take this set of markers first, run it on my training population to get my new model, and then use those to calculate the prediction of my selection population. I mean, some people would just drop the other markers, but then you're not spreading the effects across all your markers anymore. So you really, and this is, so I'm negative on genotyping by sequencing, right? Because it actually ends up creating more work for you because you don't get the same set of markers every time, right? I'm a much bigger believer in having robust markers that you can use reproducibly all the time. At the end of the day, you end up looking at something like this. Okay, so similar to what we were seeing before, we get this group that we clearly don't want. I mean, this actually opens up kind of an interesting statistical issue, because maybe when we calculate our population mean, we should actually omit these individuals and do it again. So in other words, is our mean here or is our mean here, right? No, because it affects where you're selecting, okay? But at the end of the day, we're going to use this to make our selections, and then these, in theory, go to the field, where we then collect phenotypic data, and then we can add that back into the training population.